Hi, I'm Tyler Browne. I'm a PhD student in the Department of Biochemistry at Western University. I'm going to be talking to you today about the prediction of bacterial CRISPR-Cas9 activity with machine learning. So the adaptive immune system CRISPR has shown promise as a tool for use in genetic engineering and more recently as a next-generation antimicrobial agent. In the majority of CRISPR applications, the CRISPR-Cas9 nuclease is targeted to a site by a single guide RNA molecule, an sgRNA, where it then introduces a double-strand DNA break. The success of this tool is limited by the wide variation in on-target activity of the targeting sgRNA sequence. Now, the molecular understanding of the sgRNA interactions with the DNA target and corresponding Cas9 nuclease have enabled computational modeling of sgRNA activity at desired on-targets and undesired off-target sites. In the past decade, there have been several data-driven attempts to predict the on- and off-target activity of sgRNAs used by CRISPR-Cas9. However, in testing, no correlation was found between our data and the predictions from these prior models, a clear issue in model generalization. Additionally, they tend to miss a few important features. Therefore, we sought to develop a more robust computational model for the prediction of bacterial CRISPR-Cas9 sgRNA activity. And to do this, we actually used two novel data sets, a luminous sequencing data set and an optical density data set. Both of these rely upon the testing of unique sgRNAs in conjunction with the CRISPR-Cas9 molecule to cleave a toxic plasmid and then measure the growth of the bacteria based upon that. In the luminous sequencing dataset, better cleavage from a sgRNA will result in better growth, and therefore when you sequence the bacteria, you will obtain higher read counts for each sgRNA sequence. In the optical density dataset, on the other hand, growth rate is measured by optical density change over time. Now, both of these types of data are compositional in nature, and therefore they were transformed using the Atchison mean function in CODA-seq to allow for interpretability. The input sgRNA sequences were one-hot encoded, providing the model with matrices. And by one-hot encoding, all that I mean is that the 20 nucleotide sequence comprised of, A, C, G, T nucleotides was put into a 4 by 20 binary matrix with 4 nucleotides along the 20 nucleotide long sequence. Melting temperatures for each input sgRNA sequence were also calculated using nearest neighbor thermodynamics. So if you take a look at figure 3, you can see the architecture of our neural network. The input that was used for training and validation was the sgRNA sequences and their corresponding activity values. For each sgRNA sequence, a melting temperature was also calculated. And using this, those two inputs, the sequence and melting temperature, were put into the neural network with two branches for the sequence itself and the melting temperatures. For the sequence itself, that sequence in binary encoding is passed through a few convolutional layers, then a recurrent neural network layer, and then finally some densely connected layers. On the melting temperature side, that input was just passed through a few dense layers. The outputs of these are concatenated, placed into more dense layers, from which a final activity prediction for the sgRNA is outputted. Now, in terms of how our model performed after hyperparameter tuning and validation, it performs quite well on the sequencer data. So that, of course, that's the one that it was trained on, so that's to be expected. When testing the model on the optical density data set, its predictive ability does, in fact, drop as expected. However, that being said, the other models that we tested, there were 5 other models that were tested, also performed quite poorly. In fact, they performed worse than our model, with the eukaryotic models performing the worst, actually having a negative predictive ability, and the bacterial models having approximately a predictability of about 0. So it's about as good as picking the best sequences for activity at random. In terms of our model not performing incredibly well, this isn't especially concerning to me for two main reasons. Our model is still in the final development stage with hyperparameter tuning ongoing. There's an additional off-target branch that we are still looking at incorporating, and as more data is generated, our model should hopefully improve in terms of performance on testing datasets. And secondly, it may be the case that a model needs to be constructed using optical density data to perform well on optical density data. So to display the generalizability of our model, we intend to benchmark it using the activity datasets from other bacterial sgRNA activity models. So thank you for listening to my presentation,